Go. You can go.
Okay, yes. Crisis. The world we know today is facing a crisis. Yet, before sanctions were properly imposed, many world leaders downplayed this crisis that was broadening worldwide. But what makes the COVID-19 pandemic the crisis it is today? What do we know about it thus far? How can we deal with it? My name is Jet Lim, and I'm an undergraduate student pursuing my education of biomedical engineering with a second major in innovation and design at the National University of Singapore. As I transition into my second year, during this summer break, I've been researching, studying, and following the COVID-19 pandemic for the past few months. And today, I would like to immerse all of you in a brief journey through time as we explore an informative overview of the COVID-19 pandemic, from past till present, from the unevolved to the evolved. Later, we will be examining the virology, epidemiology, and clinical features in relation to COVID-19. But first, what is the COVID-19 pandemic? It is the current outbreak of the novel coronavirus, severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2. So SARS-CoV-2 is the strain of coronavirus that causes COVID-19, as we all know, a respiratory illness. Its epicenter is located in the Hubei province of the People's Republic of China, and it has since spread to numerous other countries. Now, as the name suggests, the COVID-19 pandemic was first reported in 2019, late 2019. But what is so critical about these pathogens, the coronaviruses? Let us explore their virology. So in general, viruses, one of the first living things on Earth, they are not alive like the way we are. They have a need to take over other living cells to reproduce, and that are their goals: to survive and to simply multiply themselves. So these coronaviruses, they are enveloped positive sense single-stranded large ribonucleic acid, or RNA, and they can infect humans as well as a wide span of animals. In fact, their history dates back to the 1960s when they were first described by Tyrrell and Bino, microvirologists, who acquired the viruses from people with common colds. And so they have a unique structure which resembles spherical virions with a core shell and club-shaped surface projections that look like a solar corona. And as a result, they were termed coronaviruses, with corona meaning crown in Latin. So once a virus gets into the body, those little projections, they act as a key, locking onto the proteins present on the outside of many of these human cells. And after breaking into cells, the instructions are given by the virus to produce more copies of itself. And this possibly leads to the invasion of more and more cells, which can lead to the surfacing of symptoms in otherwise asymptomatic patients. In fact, these viruses are actually better at spreading if their hosts do not turn out to be that unwell. All right, so sorry, it's not loading. Sorry, just give me a minute.
Jet, do you need any help?
I'm not sure why my slide isn't loading.
Try to exit from the wall screen and then you can re-enter. Just push ESC.
Sorry, I think it's hanging a little.
Do you have any video in the slide? Oh yes.
Sorry, just give me a minute.
No problem.
Still problem?
Yeah, I think it's hanging a little.
Is it just the screen sharing, sir?
Yeah, sorry.
So, Jet, I can suggest you, I saw that you have just Chrome that is on your computer. You have PowerPoint and Google Chrome. That's it. Am I right?
Yes, yes.
Because usually it is the video card. So you can try to speak without your camera and check if it is working that way. So turn off your camera and try to restart the presentation and resharing the screen.
Okay, can.
Yes, I think that would be fine. Yeah, exactly.
Start to share the screen. Otherwise, Jet, you can also send me the presentation, and of course, I can share the screen for you. Let's see if it is working.
Is this currently working?
We can see your screen.
Yes, we can see your screen now, sir.
Okay, can. Thank you.
Yeah, thank you, Mr. Jetsley.
Yes, okay, can. Thank you. So I will just carry on from here.
Yes.
Yes, so actually, so their goal is just to survive and multiply. And in fact, these viruses, they are actually better at spreading if their hosts do not turn out to be that unwell. Okay, so we have 4 genuses, namely the alpha, beta, gamma, and delta coronaviruses that exist. And while alpha and beta coronaviruses appear to come from mammals, especially bats, gamma and delta viruses come from pigs and birds. And the genome size, which is the total amount of deoxyribonucleic acid, or DNA, contained within one copy of a single complete genome, it varies between 26 and 32 kilobases. And out of the 7 subtypes of infectious coronaviruses, the beta coronaviruses may cause dire disease and morbidities, while alpha coronaviruses cause asymptomatic or slight symptomatic infections. So SARS-CoV-2, the cause of COVID-19, belongs to the B viral lineage out of 4 lineages of the beta coronaviruses, and is actually quite similar to the SARS-CoV virus with a zoonotic origin, which caused the 2002 to 2004 SARS epidemic. And ever since SARS concluded in 2004, with bats being the primary host to many of these SARS-related coronaviruses, there was this group called EcoHealth Alliance that has been coming down to bat caves in southern China to catch bats and test them for viruses, subsequently noting those that could easily reach us via animal to human transmissions. And they are then labeled as high risk or low risk based on how alike they are to the viruses that have already afflicted humans. So just a few years ago, there was this discovery of a bat coronavirus, RATG13, which was in accordance with the criteria for low risk because it seemed to be too distant and it just didn't appear to be a very clear and present danger for us. But when scientists sequenced the genome of SARS-CoV-2, the virus behind this COVID-19, they found that 96% of it was actually identical to that bat virus at the whole genome level. So it is believed by researchers that this bat virus possibly evolved into SARS-CoV-2 that could infect humans. So one thing that could be drawn from this is how impossible it is for us really to predict all the ways a virus might evolve. And the bat coronavirus may have possibly just mutated in another bat, or it may have been transmitted to another species before transmission to us, such as through a snake, a pangolin, or even a fish. Now, let us take a look at the epidemiology of COVID-19, where we will examine the distribution, patterns, and determinants of health and COVID-19 disease conditions. So SARS-CoV-2 has apparently transitioned from animals to humans in the Huanan seafood wholesale market in Wuhan, part of the Hubei province of China. However, the original source of viral transmission to humans currently remains unclear, although it is known that the first infections from SARS-CoV-2 were discovered in Wuhan. So it rapidly spread, and this resulted in an epidemic throughout China, followed by an increasing number of cases in other countries and continents worldwide. The World Health Organization, or WHO, named the disease COVID-19 in February 2020, which, as we all know, stands for Coronavirus Disease 2019. And globally, more than 7 million confirmed cases of COVID-19 have been reported. As of now, cases have been reported in all continents except Antarctica, just so we are clear on that. And as the outbreak progressed, person-to-person spread became the primary means of transmission from the initial association to live animals. Based on current evidence, it is thought to occur through close-range contact, mainly through respiratory droplets, which typically do not travel more than 2 meters. So the release of this virus in the respiratory secretions when an infected person coughs, sneezes, or verbally communicates can infect someone else if it makes direct contact with the mucous membranes. And infection can also occur if one comes into contact with an infected surface and then touches one's eyes, nose, or mouth. And actually, in general, this is a problem because the average person makes contact with the face in about 16 times an hour. There are some differing sources here and there, but it's about there. And so as for the airborne route of transmission, its existence has been debated with low certainty and clarity. For the non-respiratory specimens with SARS-CoV-2, so SARS-CoV-2 has been detected in such specimens, including stool, blood, ocular secretions, and semen. But the role of these areas in spreading is currently uncertain. And it is worth noting that there have been some reports that described the detection of SARS-CoV-2 RNA from stool specimens, even after viral RNA could no longer be picked up from upper respiratory specimens. And live virus has also been cultured from stool in certain rare cases. Although it would be hard to corroborate, fecal-oral transmission has not been clinically described, and in fact, based on a joint WHO-China report, they found that it did not appear to be a noteworthy component in the spread of infection. So the finding of SARS-CoV-2 RNA in blood fluid has also been reported in some, but not all studies that have tested for it. However, the possibility of blood-borne transmission, such as through blood products or needle sticks, it seems to be low, and respiratory viruses are in most cases not transmitted through the blood-borne route. And transfusion-transmitted infection has not been reported for SARS-CoV-2. SARS-CoV, or for the related Middle East Respiratory Syndrome coronavirus, MERS-CoV. There is also a lack of evidence that SARS-CoV-2 can be spread through contact with non-mucous membrane sites, such as abrasions. When it comes to the viral epidemiology, apart from the route of transmission, we need to examine its viral shedding, which is the release of viral offspring after successful reproduction during a host cell infection, and its period of infectivity. So to date, the exact interval during which a person with COVID-19 is infectious is still uncertain, and it seems that SARS-CoV-2 can be transmitted before the development of symptoms and can take place throughout the course of illness, especially early in the course. However, it should be noted that most data introducing this issue are from studies assessing viral RNA detection from respiratory and other specimens. So this detection of viral RNA, it does not necessarily highlight the presence of infectious virus. Thus, prolonged viral RNA detection after resolving the illness does not necessarily suggest infectiousness. Some largely indirect data also suggest that infected individuals have more potential to be infectious in the early infection stages, and viral RNA levels from upper respiratory specimens, they appear to be much higher not long after symptom onset, as opposed to later in the illness. Also, there was this study of 9 patients with mild COVID-19, where they found that infectious virus was actually isolated from the nasopharyngeal, oropharyngeal, and sputum specimens during the first 8 days of illness, but not after this interval despite continued high viral RNA levels at these sites. In one study that evaluated many close contacts of 100 infected patients in Taiwan, all of the 22 secondary cases of these close contacts had their first exposure to the index case. And these patients who have already been infected within 6 days of symptom onset. However, there were no infections documented in the remaining contacts whose exposure was past this interval. So there has been some documented data on the transmission of SARS-CoV-2 from asymptomatic individuals or individuals without any symptoms or individuals within the incubation period. So from a study of a SARS-CoV-2 outbreak in a long-term care facility, the biologic basis for this is a result of infectious virus that was cultured using this test called reverse transcription polymerase chain reaction, or RT-PCR. And they were found to be positive in upper respiratory tract specimens in pre-symptomatic and asymptomatic patients as early as 6 days before the development of typical symptoms. So this RT-PCR test is for the qualitative detection of nucleic acid from SARS-CoV-2 in upper and lower respiratory specimens, such as nasal or nasopharyngeal swabs, sputum, lower respiratory tract aspirates, etc. So but the extent to which such transmission actually occurs and how much it actually contributes to the pandemic currently remains unknown. There was this Singaporean analysis of locally acquired COVID cases, which found that the transmission during the incubation period, it occurred in about 6.4% of cases. And in such cases, the exposures occurred about 1 to 3 days after symptom development. Once again, it is uncertain how long a person remains infectious, but available data does point to the lack of clear association between prolonged viral RNA shedding after symptom resolution and prolonged infectiousness. This duration of viral RNA shedding can vary with a potentially wide range depending on the severity of illness. So in one study, asymptomatic infection was associated with a higher probability of nasopharyngeal viral RNA clearance within the first week of diagnosis compared to symptomatic infection. Yet in another study of 21 patients with mild illness and absence of hypoxia, 90% had repeated negative viral RNA tests on nasopharyngeal swabs by 10 days after the onset of symptoms, with tests being positive for longer durations in patients with more severe illnesses. So as for the risk of transmission for SARS-CoV-2, it really does vary from individual to individual by the type and duration of exposure, the use of preventive measures, and possibly even individual factors like the amount of viral material in respiratory secretions. Most of these secondary infections have been indicated among household contacts in congregate or healthcare settings when personal protective equipment or PPE were not used. So these settings include hospitals, long-term care facilities, as well as various closed settings like cruise ships. However, the reported clusters of cases after work or social congregations also indicate the risk of transmission through close non-household contact. It is worth noting that the risk of transmission with more indirect contact, such as walking past an afflicted individual in public, and coming into contact with items that were previously handled by afflicted individuals, is not well established and is likely to be low. So this may explain why during lockdown or equivalent measures, some nations allowed essential roaming, but not any congregation with friends. So in light of these clusters, we know that contact tracing was implemented in early stages of the COVID-19 epidemics present at various locations, which suggested that most secondary infections were among household contacts, with a secondary attack rate of up to 15%. And based on a joint WHO-China report, the rate of secondary COVID-19 infection in various locations range from about 1 to 5% among tens of thousands of close contacts of confirmed patients in China, where most of these cases occurred within households, with an in-household secondary attack rate of 3 to 10%. So in the United States, it has been found that the symptomatic secondary attack rate was approximately 0.45% among 445 close contacts of 10 confirmed patients. And among household members, the rate was 10.5%. In other similar studies, such as one conducted in Korea, the rates were close. And for that one, I think the secondary infections was 0.55% of all contacts and 7.6% of family members. So these clusters of cases have also been described after family work or social gatherings where close personal contact can occur. So just to share an example, the epidemiological analysis of a cluster of cases in the state of Illinois displayed likely transmission via 2 family gatherings at which food and embraces were shared, and they had extended proximal verbal conversations exchanged with symptomatic individuals who were later indicated to be afflicted with COVID-19. So the possibility of a high transmission risk through singing in close proximity has also been indicated in a report of an outbreak among a choir group. With regard to the environmental contamination imposed, virus that is on contaminated surfaces or items could be another route of infection if vulnerable individuals come into contact with these surfaces, followed by a transfer of this infectious virus to mucous membranes in the mouth, eyes, or nose. So the rate of occurrence and relativistic significance of this type of transmission to date remains unclear, and there's a higher likelihood of it being a possible source of infection in settings where there already exists heavy viral contamination, such as in households containing the infected or in hospitals and other healthcare areas. So Considerable virus contamination of environmental surfaces in hospital rooms of patients with COVID-19 has also been indicated. In a study from Singapore, viral RNA was detected on nearly all of the surface tested, namely like the handles, light switches, bed and handrails, etc. And it was found in the airborne infection isolation room of a patient with symptomatic mild COVID-19, evaluated before routine cleaning. However, viral RNA was not detected on similar surfaces in the rooms of two other similarly symptomatic patients after routine cleaning with a sodium dichloroisocyanurate, which is a widely used cleansing agent and disinfectant. So once again, viral RNA detection does not necessarily confirm the presence of infectious virus. As of now, it is unknown how long SARS-CoV-2 can persist on surfaces, though other coronaviruses have been tested and could survive on inert surfaces for up to 6 to 9 days without disinfection. And according to data related to these other coronaviruses, the duration of viral persistence on surfaces also likely depends on surrounding temperature and humidity, as well as the size of the initial inoculum used for inoculation to artificially induce immunity against the viruses caused by this, against the diseases caused by these coronaviruses. So in a study evaluating the survivability of these viruses dried on a plastic surface at room temperature pressure, a specimen containing SARS-CoV, which is very closely related to SARS-CoV-2, had detectable infectivity at 6 days, but not 9 days. However, inspecting similar studies, they found that various disinfectants such as ethanol with concentrations between about 62 and 71%, it deactivated a number of coronaviruses related to SARS-CoV-2 within a minute. And simulated sunlight has also been shown to deactivate SARS-CoV-2 in about 15 to 20 minutes in experimental conditions, with higher levels of ultraviolet B light with wavelengths of 290 to 320 nanometers associated with more rapid deactivation. So we can draw from this data the importance of environmental disinfection in the home and healthcare setting. With regard to the risk of animal contact, SARS-CoV-2 infection is thought to have begun its course of transmission to humans via an animal host, but the ongoing risk of transmission via such contact is still uncertain. In fact, there is no evidence suggesting that animals, including domesticated animals, are a significant source of infection in humans. With the uncertainty about the transmission risk and the apparent susceptibility of some animals to SARS-CoV-2 infection, it was recommended by the United States Centers for Disease Control and Prevention that personal pets be kept distant from other animals or external people. And additionally, people with confirmed or suspected COVID-19 were advised to stay away from household pets and household members for the duration of their self-isolation period. To my knowledge, there have been no reported incidences of domesticated animals transmitting SARS-CoV-2 infection directly to humans. So for immunity, it is known that actually there are antibodies to the virus that are induced in those who have become infected. And initial evidence indicates that some of these antibodies are protective, but this has yet to be clearly established. Also, it is unknown whether all infected patients will naturally have this protective immune response, and if there's any lasting duration of any protective effect. So as of now, more information on protective immunity following COVID-19 are emerging. So a recent case studies evaluating experimental convalescent plasma for treatment of COVID-19, it indicated the presence of neutralizing activity with improvements in clinical status in the plasma of recovered patients that seemed to be transferred to these recipients after plasma infusion. Similarly, in another study of close to 24 patients who have successfully recovered from COVID-19, the antibodies to the receptor binding domain of the spike protein and the nucleocapsid protein were detected by enzyme-linked immunosorbent assay, or ELISA. So in most patients by 14 days following the onset of their symptoms. And these ELISA antibodies, they actually correlated with neutralizing activity as well. Animal studies as of date have suggested that the immune response to infection could possibly offer some protection against reinfection, at least in the short run. Additionally, studies have also identified SARS-CoV-2 specific T cell, which is a type of lymphocyte, responses in patients after recovering from COVID-19 and in individuals who had accepted an investigational SARS-CoV-2 vaccine, which suggests the potential for a durable T cell immune response. So some of these studies have highlighted positive RT-PCR tests for SARS-CoV-2 in patients that have been confirmed with COVID-19 after a clinical improvement and negative results on 2 consecutive tests. However, these positive tests that were indicated shortly after the negative test, they were not associated with worsening symptoms and has improbable representation of infectious virus and probably did not illustrate reinfection. So with regard to infectious spread, a report from the Korea Centers for Disease Control and Prevention of patients afflicted with COVID-19 who had a repeat positive RNA test, despite being previously cleared of infection from their isolation, the infectious virus could not be isolated in cell culture in any of the patients tested. Also, there were no newfound confirmed cases that were traced to exposure within the duration of the repeat positive test. So as a result of all these settings, the United States Food and Drug Administration has authorized the emergency use of tests that identify these antibodies against SARS-CoV-2 in serum or plasma. So hopefully, if evidence does confirm a protective immune response from the presence of these antibodies, serologic screening will be important in understanding population immunity and identifying those people with a lower risk for reinfection. And now we'll be moving on to some of the general clinical features presented. So the initial clinical sign of SARS-CoV-2 related disease, COVID-19, which permitted case detection, was actually pneumonia, which is an infection of the unilateral or bilateral lungs. And various recent reports also indicate gastrointestinal symptoms and asymptomatic infections here and there. In general, the spectrum of asymptomatic infection, it can range from mild to critical, with most infections not being severe. Severe illness can occur in healthy individuals of any age, but it mainly occurs in older adults or those with underlying medical comorbidities. For age, adults who are middle-aged and above are most commonly affected, with older adults having possibly more severe disease and increased mortality. So symptomatic infection in children, it appears to be relatively uncommon. When it occurs, it is usually mild, although severe cases have also been reported. So some of these concomitant conditions that have been associated with severe illness and mortality for SARS-CoV-2, for COVID-19, include cardiovascular diseases, diabetes, high blood pressure or hypertension, chronic lung or kidney diseases, cancer, obesity, and smoking. All right, so as for the clinical manifestations on initial presentation, pneumonia seems to be the most common grave manifestation of infection, indicated mainly by pyrexia or fever, cough, dyspnea, or shortness of breath, and bilateral infiltrates of the lung, which these infiltrates, they linger within the parenchyma of the lungs on chest imaging. However, some other features such as diarrhea, upper respiratory tract symptoms, muscular pain or myalgias, and olfactory or gustation disorders, they are also frequent. So thus far, there are no precise clinically presented features that can yet reliably differentiate COVID-19 from other viral respiratory infections. Although the presence of dyspnea does several days post onset of initial symptoms does or could possibly suggest that. And according to a study of patients hospitalized with a pneumonia due to COVID-19 in Wuhan, the most common clinical features of at the onset of illness were fever in about 99% of cases, fatigue in 70%, dry cough in 59%, anorexia in 40%, myalgias in 35%, dyspnea in 31%, sputum production in 27% of cases. But it should be noted that actually fever is not a universal finding on presentation when we corroborate with other studies, though these studies have indicated a similar scope of clinical findings on presentation. So some other reported symptoms from studies will include a conjunctivitis, headache, sore throat, and rhinorrhea, or what is commonly known as a runny nose. Sometimes we hear of people being issued stay-home notices of 2 weeks due to the incubation period for COVID-19, which is also known as the pre-symptomatic period, where some infected people could be contagious and transmissions can actually occur. So it is thought to be within 14 days following exposure, with most cases actually happening about 4 to 5 days post-exposure. So some of the complications of COVID-19 have been indicated to be acute respiratory distress syndrome, or ARDS, arrhythmias, acute cardiac injury, and shock. It has been stated by the World Health Organization that for the recovery time, it seems to be approximately 2 weeks for mild infections and about 3 to 6 weeks for severe disease. So today we are all fighting against the COVID-19. From my experience, the Biomedical Engineering Department of the National University of Singapore is participating actively in the fight, with some of my professors working on various areas such as fighting infectious diseases with artificial intelligence and modeling the spread of COVID-19. My family, on the other hand, is also participating actively, ensuring that we wear masks when we are outside, sanitizing regularly, and washing our hands often and appropriately, amongst many other things. So if there's one thing you can take away from this, it is that I think we all do have a part to play, and every one of us can contribute, be it helping out in academia in this boot camp, or even just following the sanctioned rules. So speaking of rules, I would like to share a quote by Mr. Gan Kim Yong, Minister of Health in Singapore. Which goes, you can fool the rules, but you cannot fool the virus. Let us turn the battle around together. Thank you. Are there any questions?
So congratulations and thanks for the presentation, Jet. It was really interesting. I think also full of inspiration for those people who are doing the project on
Yes.
diagnosis.
Thank you.
So Terence has a question.
Yes.
Thank you, Camilo. Jet, I wanted to ask, you mentioned Antarctica region has not recorded yet a case. Is it a case of climatic conditions, or it's a case of regulations that are put into place to minimize this?
Oh, the Antarctica one, is it? No, it's because it's currently not properly inhabited. by people, I believe. So as far as I know, I don't think anyone say purposefully lives in the Antarctica and actually contracted COVID in Antarctica. Makes sense. As in there are no nations on Antarctica. So whereas on other continents there are other nations. So actually sometimes I see news reports that states there's a confirmed cases reported in all continents, but the truth is that we have to remember that Antarctica is also a continent, right? And there's actually no nations on it, no hospitals, no people with a national identity tied to it. Does that answer your question?
Yes, it does.
We have also a question from Lynette.
Hi, thank you for the presentation. You've answered like most of the questions that I actually had with regards to immunity and all of that. But I just wanted to, you did mention that SARS has, like COVID-19 has genome similarity of 96% with another virus. I can't remember which one you mentioned.
Yes, correct. To SARS-CoV, yes.
Yes. So my question is, with the COVID-19, has there been any significant mutations noted in different patients that have been diagnosed already? Have they noticed any significant?
Sorry, could you repeat?
Have they? Sorry. Yes. I want to know if they have done any. Isn't they have done sequencing? Have they noticed any significant mutations and variations within the COVID-19 itself? Because with viruses, you know, yeah.
Actually, I think the last I read, they haven't really noticed much about it. But the reason I think why they feel that it's a potential threat is because, you know, because just a few years ago, I'm not sure if it was 2012 or 2013 or something, when they found this bad coronavirus, RATG13, right? Yeah, so yes. Then afterwards, when COVID, so initially they labeled it as a low risk because like it just seemed to have that kind of sequence that shouldn't be impacting humans at a large scale. And when COVID actually happened, and then we went to sequence this, they found that there was this a 96% similarity, and it just suggests that virus, that RATG13 virus, actually mutated into the COVID. The SARS-CoV-2 virus it is today. So actually, I think currently large mutations, no, there have been none reported so far as I know. Yeah.
Okay.
But I don't think it takes a very large mutation to really cause an impact for that matter. Yes.
Okay, thank you.
No problem.
I have a question, actually. You mentioned a number of fluids of the human body in which the virus was identified. Do you have any evidence or paper to suggest in which virus or particle of the virus were found in sweat?
In sweat, I think for sweat, I have not come across it yet. Yeah, currently I do not have.
Okay, now this can be important also thinking about all the people who want to go back to do sport. Okay, Susan.
Thanks, Camelo, and thanks for a very informative presentation. Now, my question relates to the issue of temperature screening. And from your presentation, there are a couple of things that have struck me, like first is the asymptomatic nature of infections in children.
Yes.
So essentially that says that children would really be a dangerous lot when it comes to transmission, since we really can't pick it out.
Yes.
Then the other point that you also said was that temperature is not an indicator in most of the studies, in as much as it is also associated with pneumonia, because pneumonia-like symptoms are associated with COVID.
Sorry, could I just clarify? I think,
Yeah.
no, actually, I believe that according to this data, it actually does state that the duration of the viral persistence on surfaces, okay, so when we talk about environmental contamination, it could depend on the surrounding temperature, actually.
Okay, I'm talking about fever, not
Okay.
environmental
okay.
temperature.
Okay.
Yeah.
All right.
So in your opinion, then what is the role of the almost mandatory temperature screening processes that have been installed in a lot of the public spaces? To get into any public space nowadays, you must accept a temperature check. What's the role of doing that?
Okay, because I believe that okay, so actually most studies have found that high temperature like fever, pyrexia, it occurs in sorry. Yeah, it occurs in quite a number of cases, and actually there are certain studies that have corroborated this finding. But I think ultimately, yes, it may not be absolute certainty, but I think the role of actually making sure that we do check on all of this fever and stuff is also because some studies have reported fever to be one of the most common clinical signs. And even though it may not be with 100% certainty that just because one doesn't have fever doesn't necessarily mean one doesn't have COVID, I think it's a very good safeguard just to make sure that at least they can try to test, you know, to prevent a clustering of cases, maybe to even test people with fever, etc., to aid in the contact tracing implementation. Yeah, that's my take on it.
Okay, thanks.
No problem.
So if there are no more questions.
Yeah, I have one question, Camelo. Before
go David.
Yes.
Before Dr. leaves, you talked about the early detection of COVID-19 to be similar to that of pneumonia. Did I get that right? The slide which you posted
no, as in the early clinical sign is pneumonia.
Is pneumonia.
Yes.
So if at all I can get initial clinical signs of COVID-19 is pneumonia. So if I can use images of X-rays to detect pneumonia, can I use X-rays images to differentiate between the pneumonia and COVID-19?
Oh, okay. So this is the problem with it, in that actually even in cases where there's a pneumonia, it does not necessarily mean that one necessarily has a COVID-19. So as of now, I know that there are no clinical signs, okay, on presentation, like just normal signs and symptoms brought in by patients to the doctor, for instance, that can immediately let the doctor know that this person is suffering from has COVID-19. So they still have to go through the nasopharyngeal swab testings, etc., to find out. Pneumonia itself is not a direct and absolute indicator. In fact, there are no symptoms or signs as of now that are direct indicators of COVID-19. Yeah.
Okay. Thank you, Camelo.
Thank you.
Thank you, David, for the question. Other question from the audience?
Hi.
Who is
Just
speaking?
I want to Okay, to just touch through clinical presentation. First of all, thank you for this very comprehensive presentation and very informative. Thank you very much. I want to touch about that COVID-19 may present with other presentation which are uncommon, and sometimes someone may not think this may be COVID-19, like some patients present with neurological symptoms, and some of them present with heart attacks and acute abdominal symptoms, which may be sometimes bizarre. One other interesting symptom, which is reported in about 2/3 of patients in UK, which is loss of smell. Just these are just points I want to point to.
Okay.
Thank you.
So I just would like to add like my personal experience to the evaluation of temperature. I think that the evaluation of temperature is a clinical parameter. It is better that there are healthcare providers in taking the temperature because they know actually how to manage and how to consider the data. I was going to a shop, and it was so hot outside, and I was running with my car. So temperature was on my head at 39.2. So if there were someone not skilled, this can generate an alarm. Instead, the lady, was a professional lady, asked me to wait for 5 minutes. I just came back to the normal temperature of 36.2.3. So screening with temperature, or even with pulse oximeter, is perhaps dangerous. So that is personal experience if it is not done by professionals. I'll stop the presentation, and